So here I've just given you some food for thought. Here's an image of embryonic development. Remember that all of us develop a single fertilized egg, one cell, right? And all of our organ systems that are composed of thousands of cells, the body probably has about 1 trillion cells in a typical adult, okay, come from that one cell, right? So think about all of the regulation that has to happen to get that one cell to be a liver cell, a heart cell, a skin cell, a rod cell in your eyes, or whatever. All the cells that we started the semester mentioning on that first day that we met, right? What scientists used to think is that each cell type had a different set of DNA, and that's why it looked different. But what we now know is that we all of our cells, with a few exceptions, have exactly the same DNA content. So it's really the control of how the cell uses that information that determines what kind of cell type it is. Chapter 8 goes over the types of genes, housekeeping genes or specialized genes, the regulation, and really it focuses on the regulation of that first step in the central dogma, the transcription. Then it talks about some other ways of regulating DNA accessibility by adding groups, by acetylation to kind of open the condensed form of DNA. And we already talked about a couple of ways that after transcription there's control, kind of those checks and balances of does all the RNA, has it been processed to the mature form? The splicing can happen differently in different cells, producing eventually different proteins, and modifications that occur to the protein after translation, which are not limited to phosphorylation, acetylation, and ubiquination, but can also include glycosylation and other modifications. But really, when we look at really any cell, but here in your book they've given a very dramatic example of a neuron and a white blood cell. Extremely different when we look at these cells. For any of you who have looked at, who have taken anatomy and physiology, you've looked at a lot of different tissues and they all have certain characteristics, right? But they're all from the same type of organism. So even within your own body, your white blood cells are very different in how they appear versus your neurons or your skin cells. So what accounts for that difference? We know from a karyotype, we can look at these cells and find the same set of chromosomes in each of these cells. So what is it that makes them so different on the outside, which we call the phenotype? Right? And that's the regulation of what genes are used. So we have about 22,000 genes as humans in our genome. Cells don't ever use all of those genes at one time. Instead, they use a subset anywhere from several thousand, maybe 5,000 to 15,000 of those genes are used in different cell types. I think it's around 5,000 to 15,000. There are simpler cells which, but for the most part it's around, on average, 10,000 genes that are being used out of those 20,000. Some of those genes are the same set of genes that you find on in almost every single cell. Okay, so any cell that's carrying out metabolism needs to make what in order to carry out those uphill reactions? ATP, right? So certain genes are housekeeping genes. They're just constitutive, they're always on, they're always made. They're common between a neuron and a white blood cell. But a whole nother set of genes are specialized to that cell type. So how do we know, now that we are in the molecular era of science and biology, how do we know that each of our body cells carries enough information to make any other cell type? It was a question, but I'm going to answer it for you. So,
you said he's going to answer.
Yeah. So it's rhetorical. So it's by cloning. So we know that if you take an adult frog and take a skin cell, just one skin cell from adult frog, take out just the nuclear material, so just that nuclear compartment, put it into a frog egg that doesn't have its nucleus anymore, for the most part, you're going to get another little tadpole that develops in a perfectly normal frog. Okay, so that tells us that that skin cell has all the information it needs to make a new frog. Now we know it doesn't happen that way because the skin cell is what we call differentiated. It's already kind of gone through life, it's gone to college, decided it wanted to be a skin cell. It's not going to change from a skin cell. It's programmed to be, it's been educated to be a skin cell. So it's differentiated. Question? Do they reproduce? They could. Yep. So this is like IVF in vitro fertilization. This is basically what they do if there's something wrong with the father's sperm that they can't normally, naturally conceive. They can use the DNA from his sperm to fertilize an egg just in a petri dish in the lab and make another human being that can reproduce when they become an adult. Yeah. And it's been done with plants as well. Just a single cell from a carrot can be used to grow a new carrot plant, an entire plant. So we know that these cloning, whether it's in a amphibian like a frog or a plant or even Dolly the sheep, or this kind of in vitro fertilization in cows, this can be used to produce an entire new organism from the DNA that was contained in whatever the donor cell is. So we know the information is there, but we still have the question of, okay, what makes all of these cells in the calf different from each other? This calf also has the skin cell and the neuron and the liver cell and all of these different cells that make up the organs, the tissues and the organs that make up the organism. So there are a lot of ways that gene expression, whether it's constitutive genes or regulated genes, there's a lot of ways that they can be controlled. In the case of constitutive genes, they're the ones that are always on. Okay, so we're really going to focus on those specialized genes, that subset of the genes that make cells different. So they are regulated. Some are turned on, some are turned off. So some end up making protein, others don't. So these regulated genes are what make different cell types different. Okay, they're cell type specific. They have a different profile of genes that are expressed in my skin cells than in my liver cells. So you can either measure it on the level of messenger RNA that's present in these cells is different, or proteins in these cells are different. And as I mentioned, some are in common for those constitutive genes. But I'm just going to focus on those regulated genes for the rest of class today. There are also times, being that we are living organisms, and part of the definition of life is that we can respond to change, that cells need to be able to respond to changes in their environment. So our hormones signal a change. Maybe we're in between meals, and so we get a signal from cortisol, the hormone that would trigger the liver cells to make more glucose so we don't faint in between meals, right? So whether they are helping with the development and differentiation of a cell into a particular type, or whether they're helping a cell that's already differentiated respond to its environment, these are all regulated genes, and they're regulated in very similar ways. So if you think about how we could regulate genes, there are several points along this central dogma of the conversion of DNA to protein, several points at which the cell can control what's there and what's not. Okay, it can just break down the protein. Okay, we don't need it anymore, let's degrade it. We have an mRNA, but we're not going to translate it, so we just don't get any protein from that. Or we have an mRNA in the nucleus, but we're not going to bring it to the cytoplasm where it can get translated. Or we have an RNA that's a pre-mRNA. Okay? We're not going to process it. Or we have a gene, we're not going to transcribe it. So, put on your thinking caps. What makes sense? What do you think is the most common point of regulation, no matter what the cell type, where it starts? Okay? Anyone? So transcriptional control. So the most common place to regulate gene expression is indeed at this first step. If you don't need it, don't transcribe it. Why do you think that makes sense? Think about the second law of thermodynamics. Energy efficient, okay? Each of these steps is taking energy. If you go all the way to transporting that message out of the nucleus and then you don't need it, well, you've just wasted all that energy. So the cell wants to do the most energy efficient activity possible because it's not that easy to make the ATP. Right? It's fighting all that entropy, that disorder, that natural trend toward disorder. Okay, so Chapter 8 is focusing on this transcriptional control. And so let's just go again and review transcription for a moment. So transcription requires RNA polymerase, has to bind to that region in the DNA, which you should also know how to draw kind of the general region of a gene, including the upstream region where you find the promoter, Tata box, that the sigma factor or general transcription factors are recognizing to help the RNA polymerase bind. RNA synthesis begins by opening or breaking that double helix, so it can read the Which strand? Coding or template strand? Which strand is the RNA polymerase reading? Template. Okay, so 3' to 5' is the movement. And you're synthesizing messenger RNA into 5' or whatever RNA it's making, if it, in the case of ribosomal RNA, whatever the gene is encoding, 5' to 3'. Okay, so you got your growing strand, and then you have your terminator, a stop site where the RNA polymerase falls off of that double helix. The double helix has now reformed those hydrogen bonds that maintain shape, and the message or the RNA molecule that's produced is released. So in prokaryotes, sigma factor is the transcription factor that helps with transcription. In eukaryotes, there are a whole collection, A through H, of general transcription factors that help RNA polymerase do its job. But in general, there are other transcription factors that you will find in some cells and not in other cells. So transcription factors are the key regulator proteins that bind to the DNA and turn on or off the transcription of that gene. Okay, so these are DNA binding proteins. Here in this figure, the blue is the double helix of the DNA, the red is the protein, and you can see it has in yellow some alpha helical regions of the protein that are interacting with the DNA. They usually interact in the major groove of the DNA, that big space, so they can get access to binding what part of the DNA, the backbone or the nucleotides. Okay, so 2 votes for backbone. Okay, so if transcription factors bind the backbone, where are they binding? Anywhere in the DNA. Do transcription factors bind anywhere in the DNA? No. They have to be specific for that gene that they're regulating. Or maybe a few genes that have that proper upstream sequence of about 10 or 20 nucleotides usually. Transcription factors need to recognize at least 10 nucleotides, as many as 20, in order to bind specifically upstream of a gene and regulate that gene's expression. So it's binding in non-covalent ways, so the non-covalent interactions that we talked about, like hydrogen bonding, van der Waals interactions, electrostatic charge-charge attractions, hydrophobic interactions. All those interactions are important for binding to a specific pattern of nucleotides. Okay, so not just any nucleotides. And so we have lots of different transcription factors in our cells. Not every cell has all the transcription factors. Each cell has its own set so that it can regulate which genes of those regulated genes are transcribed. Yep, question? Right, so where does the information for the transcription factors come in the first place? From genes in our genome. Exactly. Right, and so at the end of the chapter it mentions epigenetics, in which differentiated cells kind of pass on as they reproduce. They pass on certain patterns of modification, these covalent modifications like methylation, acetylation, or phosphorylation that will determine which areas are accessible and not accessible, that will help to determine the profile of transcription factors that are present in those cells, and kind of its inherited pattern of modifications from a differentiated cell, which then, a skin cell divides and makes a new skin cell. It doesn't make a nerve cell, right? So there are other factors, certainly. It's not exclusively transcription regulation that's at play here. It's quite complex. But I'm just trying to water it down and have you focus on just the transcriptional control for now. So the general transcription factors, as I mentioned, and we talked about a little bit last week, they are a collection of, in the case of making message, TF2, transcription factor 2 family of proteins, A through H, that start with the binding of TATA binding protein to the TATA box upstream in the promoter region of a gene, which then, so this TATA box is conserved, it's found in almost every promoter, every gene. So this is a conserved kind of general process that helps RNA polymerase to attach to the DNA at the right place so it can proceed with polymerization of those bases to make the RNA. So the first step in this general control of RNA polymerase is the TATA binding protein binding to the TATA box. That recruits TF2D to the complex, which then recruits 3 other transcription factors. And once that complex is formed, RNA polymerase 2, and that's the one I want you to remember, although there are 3 in eukaryotes. But RNA polymerase 2, it very efficiently binds. So it may kind of randomly find the right place in the promoter, but here it's much more efficient, so you got much better transcription for that gene. Okay, transcription begins. And here from this ribbon model, you can see, again, the orange and the red is the DNA. Here in the gray, you can see the base pairing. So thymine, adenine is the TATA box. As I mentioned earlier, the structure, or maybe I didn't, but just remember that the structure of many of these DNA binding transcription factors, whether they're specialized or general ones like this TBP, is often a dimer. A dimer having that quaternary structure of 2 subunits coming together, which gives them twice as many places to hold on to the DNA. So much better adherence to that site in the DNA. Better binding. And so here 1 subunit is in green, 1 subunit is in blue. And you can see they have a similar ribbon and sheet structure. So it's a homodimer. Similar subunits coming together to form 1 mature functional protein. And here you can see a similar thing in the 3-dimensional model. So the stick here, the orange is the backbone of the DNA, the bases are in purple, and the TATA binding protein is in white here in this space-filling model. So it's really kind of grabbing on and hugging that DNA, and you might have noticed that it's actually, it's actually bending, right? It's changing that conformation of the DNA, which is important. So these transcription factors, whether, an example being TATA binding protein, change the DNA structure at that site in order to help to facilitate transcription at that site.